Hi, my name is Aaron Mayfield, and I'm studying the delivery of neurotrophic factors, the hematopoietic stem cell-derived macrophages, to the brain to protect against sites of dopaminergic neurodegeneration and Parkinson's disease. In short, we're using blood-producing stem cells as vehicles to carry chemicals to the brain. The chemicals that we carry to the brain are called neurotrophic factors, which are proteins involved in maintaining and keeping up neurons. Parkinson's disease itself targets a specific area of the brain called the substantia nigra. In the nigra, there are neurons that produce dopamine, known as dopaminergic neurons. So Parkinson's disease actually kills these neurons. Without dopamine, patients can experience muscle loss as well as some loss of involuntary functions such as body temperature and behavior and problems associated with chemicals in the brain. So this first graphic here shows the comparison between a normal brain and a Parkinson's affected brain. In the normal brain, there are a considerably larger amount of neurons versus the Parkinson's brain. So this just proves that Parkinson's does actually eliminate or kill these neurons. So to do this, to overcome this problem, we send the neurotrophic factors to the brain. But if we injected the neurotrophic factors straight into the bloodstream, they would not be able to reach the brain because the body would not allow them to cross what's called the blood-brain barrier, which separates the circulatory system and the brain. So we hide them inside of stem cells, which then differentiate into macrophages, which can cross the blood-brain barrier and go to the brain. To do this, we use a lentiviral vector, which infects the stem cell with the neurotrophic factor. We genetically modify the lentiviral vector in two different ways. First, we put in the neurotrophic factor. In this case, we use GDNF. And second, we put in a macrophage-specific promoter, which only allows expression of GDNF, or the neurotrophic factor, if the stem cell differentiates into a macrophage. This is because stem cells can differentiate into many types of cells, some of which would not benefit from the acquisition of GDNF or other neurotrophic factors. So the results were very, very successful. The macrophage-specific synthetic promoter only allowed expression of the GDNF, or the neurotrophic factor, in macrophages. Here, this bar represents macrophages, and this represents other cells, and the height represents the expression of the neurotrophic factor, which was considerably higher in macrophages. The neurotrophic factor itself proved very effective for preserving dopaminergic neurons. So here we have the Parkinson-infected brains, and here we have the unaffected or control brains. This column represents the control, where the brain was untreated, and this column represents the neurotrophic factor. And you can see here that with the neurotrophic factor implemented, the dopaminergic neurons and pathways were actually preserved. In the same scenario as over here, with the Parkinson's-infected brains versus the control, and the control versus the neurotrophic factor preserved a lot of the neurons versus the control. In conclusion, using lentiviral vectors as vehicles to implement neurotrophic factors into stem cells, which then derive into macrophages to go to the brain and protect against Parkinson's disease, has proven very effective. However, we want to take this a step further. So next year, I will be working in Dr. Simlin Lee's laboratory, and we will be using a similar method. In this old method, however, the lentiviral vector would go inside of the stem cell and go inside of its nucleus and put its instructions for the neurotrophic factor somewhere randomly on one of those 23 chromosomes. Now, why is this a problem? Because if it puts its instructions on part of a chromosome that is important, that say codes for cancer or an oncogene, then it could actually rewrite those instructions and end up causing cancer. So to eliminate this, next year we'll be using the CRISPR-Cas9 technology. And this will allow us to pick the specific chromosome that we want to implement the neurotrophic factor in, so that we have a safe harbor to put the neurotrophic factor in play. In addition, we would also like to expand our studies to possibly other neurodegenerative diseases such as Alzheimer's disease. Thank you very much, and email if you have any questions.